guys, my name is Allison Hill. I am in Physiology 1010 and the date is August 16th, 2020. My topic is in anatomy and enzymology of fat absorption. I chose this topic because I am a dietetic major and the digestion and absorption of fat really interested me, and I learned a lot of new things and I'm excited to teach you guys as well. So I'm going to go ahead and begin, and I'm going to screen share my PowerPoint slide with you. Like I said, my topic is the anatomy and enzymology of fat absorption. We'll start with the route of travel of fat through the body. 90% of dietary fat comes from triglycerides. The rest of it comes from cholesterol, phospholipids, and long-chain fatty acids. Absorption starts whenever you consume food through the mouth. This is where mechanical digestion and lingual lipase happens. The food then travels down the esophagus and into the stomach, where it's mixed with simulated stomach fluid and it becomes chyme. It then travels down to the small intestine. This is where a majority of digestion occurs, and then it travels down to the large intestine, where microbial fermentation of undigested food happens. The small intestine is lined with specialized epithelium that absorbs nutrients from fat. The problem with fat absorption happens because most fat is not water soluble. The body converts any calories that it does not need into triglycerides, which are stored in fat cells. You can see a picture of a triglyceride at the top right hand corner, and it's made up of glycerol backbone plus 3 fatty acids attached to that. These triglycerides are hydrophobic, meaning they are water fearing, and they're not water soluble, meaning the body cannot break them down any farther because the body is mostly made up of water, as we all know. This is where the role of bile salts comes in. So bile salts from the liver are amphipathic. This means that bile salts have both hydrophobic and hydrophilic properties. Hydrophobic means water-fearing, hydrophilic means water-loving. So if you look down in the bottom right-hand corner, you can see a picture of a bile salt in the very right, and you can see one side is hydrophobic and the other side has green dots, which means it is hydrophilic. This allows fat to be broken down further because the hydrophobic side binds to the fat and the hydrophilic side is able to be water soluble. This emulsifies and solubilizes fat globules. From here, those fatty droplets are formed in an aqueous environment. This is then where the small intestine then breaks fat molecules into smaller, more absorbable pieces. So the small intestine contains enzyme called lipases that are able to do this, and the lipases function in an aqueous environment as well, meaning those fats that we just broke down in the last slide are hydrophilic. At the bottom, you can see a picture of a micelle. The hydrophilic heads are on the outside and the hydrophobic tails are on the inside. These micelles form and have greater surface area upon which lipases can function. This micelle is made of monoglycerides and 2 fatty acids. This is where fat absorption happens. So once that micelle makes it to the small intestine, the monoglycerides and the fatty acids inside the micelle are able to diffuse freely into the cell via simple diffusion. The cholesterol isn't so lucky. It actually is not hydrophilic, so it is transported across the apical membrane via a transporter. It's not able to diffuse freely, so these things break up and they meet up with the monoglycerides and fatty acids inside of the cell. This is where the triglycerides reform. So once the triglycerides reform, they start to form something called a chylomicron. So triglycerides and cholesterol are packaged into the core of lipoproteins for transport. So you can see here in the middle, on the right, this chylomicron has triglycerides and cholesterol. On the inside, you can see it's made up mostly of triglycerides, 75%, but cholesterol is in there as well. On the outside, you can see it's made up of proteins and phospholipids. The problem is that these chylomicrons are way too large to get into the bloodstream through capillaries. So in the last slide, at the bottom here, you can see this red tube is called a capillary, and then this green thing is called a lacteal. Most foods are able to enter the capillary, but since these chylomicrons are so large, they actually pass through the capillary and go straight into the lacteal. Once the chylomicrons are inside of this lacteal, then they can be absorbed straight into the bloodstream. Obviously, this is a longer route than most foods, making fat absorption very interesting and kind of complicated. Although the absorption of fats is pretty complicated, fats play an important role in the body, and it's very important to get as many healthy fats as you can into your diet. As a dietitian major, I want to stress this as much as possible. There are 2 types of fats. One is unsaturated fats, which are the good fats, and the other is saturated fats, which are a little more unhealthy fats. A little bit of saturated fats is okay, but you want to eat mainly unsaturated fats. The benefits of unsaturated fats are whenever they're absorbed into the bloodstream, they can be used for energy, cell repair, and growth. And your lymph system actually absorbs fatty acids to help fight an infection. This picture that I inserted on the left, you can see it just highlights a couple more benefits, such as absorption of vitamins such as A, D, E, and K. It helps regulate body temperature. It helps your immune system function properly, and it helps create a feeling of satiety or fullness. On the right, I included a picture of some healthy fats, such as avocados, fatty fish, spinach, nuts, seeds, and legumes. And that is the end of my presentation. I hope you found this informative, and thank you for watching.